In chapter 16, we are going to talk about the mechanisms of genetic variation. This is a large, quite extensive chapter, and that's the reason that we have divided this lecture video into two parts. In the part one, we are going to talk about uh, just a few different mechanisms of the DNA damage and the ways that those DNA damages carry forward the mutations, sometimes how they can be reverted or suppressed as well. And at the protein level, what are the outcomes? And then we are going to discuss in 16.3 how the DNA damages can be repaired. It's a very important chapter in terms of evolution of the microorganisms because there are many of the microorganisms which are pathogenic, and this basically the mechanisms of genetic variation is actually impose a huge amount of pathogenic challenges from viral different types of these variants that we are actually seeing is due to these mechanisms of genetic variation. But mainly we are going to talking about here the bacterial mechanism of genetic variations, and which can lead to rise to many of these antibiotic resistance. And it is of immense importance at this point. So in the part 1, 16.1 section, we are going to talk about that the mutations, which is basically defined as inheritable changes in a genome. Now, before we start, I would like to just introduce just the structure and of the nucleotides and the DNA. And this is basically a prerequisite material. And many of us actually do know these things. And this is just a primer, and it will just help us just to understand next few slides and the section. So let's take a look at that how the DNA is made and what are the different components of it. So the first thing that when we talk about a nucleoside, the nucleoside contains a pentose sugar, which does have 5 carbons, 1, 2, 3, 4, and 5. So this is a 5-carbon sugar, pentose, and depending upon whether it is a DNA, in the DNA, this 2' end, or this the second carbon, contain hydrogen only, and we call it as a deoxyribose sugar. On the other hand, in the RNA, this contains an OH, and we call this as a ribose sugar. So the sugars, both are pentose sugars, are different in the DNA and RNA. Deoxyribose, this oxygen is just only gone. And then to the first carbon, there is a glycosidic bond and a base is attached. And these bases could be either purines with a double ring and the pyrimidines, which are the single ring. And in RNA, you find uracil instead of the thymine. Now, from nucleoside, let's take a look at the nucleoside monophosphate, which is added right here to the 5th carbon, the first phosphate, so PO3 minus right here. Then the nucleotide diphosphate is with the second phosphate, nucleotide triphosphate is with the 3-phosphate, and we call this as an alpha, beta, and the gamma phosphates. Now, in the DNA, only the base, the sugar, and 1 phosphate is incorporated. So it's basically in the nucleoside monophosphate form. So on the other hand, what happened that when you have the raw material is always in a triphosphate form, and we call them as a nucleotide. So that's basically the nucleotide, and this is basically a base, sugar, and the triphosphate. Now this beta and the gamma phosphate, these 2 and 3, these are spent up in the formation of the phosphodiester bond, and only the 1 phosphate actually is present in a monophosphate form in the DNA or in the RNA. So if we just look at the structure of the DNA, and these are the nitrogenous bases, you have the sugar backbone and the base pairs are stacked upon each other in the middle portion. And how they are bonded, you can see that right here is a sugar phosphate backbone of one of the chain that goes from the 5' direction to the 3' direction, because the reason is that this phosphate is at the 5' and this OH is at the 3'. So 5' and this OH is at the 3'. So this direction is 5' to 3'. On the other hand, this is an anti-parallel, whereas you see the phosphate right here of which is bonded with the sugar. This is at the 5 prime, and then right here is the 3 prime, which is inverted in compared to here. And this particular DNA strand is going from this direction to this direction. So these are the two sugar phosphate backbone of the two DNA chains. And these are the bases, and a purine will always bond with the pyrimidine. And let's take a look at the purines. So when we are looking at the purines, one of them is adenine, and you can see that there are double-membered ring, and we numbered the rings like this. You have 1, 2, 3, 4, 5, and 6, and then 7, 8, 9. And the R group, which makes the glycosidic bond with the first carbon of the pentose sugar, forms at the 9th carbon right here. Whereas it's the same thing for the guanine, it is also right here. This is actually makes the glycosidic bond. On the other hand, for the pyrimidines, you can see that cytosine, uracil, or thymine, uracil in the RNA, the glycosidic bond is formed at the number 1 position. So we name them is at 1, 2, 3, 4, 5, 6. So the glycosidic, that number 1 is right there. So if you're just trying to see, say, look at the thymine, that how they are bonding right here. So think about that. This is your deoxyribose sugar. This is your number 1 carbon, which is number 1 carbon right here. And this is your number 1 carbon in the thymine right here. This one is right here. So what are the carbons? You have a 1, 2, 3, 4, 5, and 6. So here you can see that if we are taking just right here is a thymine, and let's take a look at the thymine right here. Where is the hydrogen bonding carbon positions are happening? And that is what is basically mentioned here. So in the thymine, you can see it is 1, 2, 3, and 4. So 3 and 4 actually hydrogen bonds. On the other hand, if we just look at the adenine to which it is bonding, you can see that it's inverted. So this is your number 1 carbon, 1, 2, 3, 4, and 5 of the pentose sugar, and this carbon is the 9th carbon. So basically, if we are looking at the adenine right here, and in the adenine, the bond is forming in between 1 and 6. So the numbers are like this: 1, 2, 3, 4, 5, 6, and then 7, 8, and 9. So right here, this is your number 1 carbon. This is your number 6 carbon, 1, 2, 3, 4, 5, and 6. So you can see that adenine 1 and 6, the bonding are forming. So now if we just take a look at the guanine, and let's take a look at the sugar, you have 1, 2, 3, 4, and 5. So this is your number 1, and that is bonded with the number 9 carbon of the guanine. And if we just looking at the structure right here and the number of carbons where the hydrogen bonds are being made, see that this is the first carbon. So you have this NH is the first carbon, and we go like this with the numbers. So it is 1, 2, 3, 4, 5, and 6, and then 7, 8, and 9. So you can see that your the guanine, you have a 1, 2, and 6. So number 1, 2, then 3, 4, 5, and 6. These are the 1, 2, and 6 are forming the hydrogen bond. On the other hand, if we just go to the cytosine right here, you can easily see that these are the 1, 2, 3, 4, 5. The number 1 carbon of the sugar is attached with the number 1 carbon of the cytosine right here. And the way we basically write that down, you can see that the 2, 3, and 4 are basically making the hydrogen bonding. So you have right here, you can see that this is number 1. So we count like this: 1, 2, 3, 4, 5, and 6. So right, cytosine, you have the bonding, and then you have the 2, 3, 4, 5, and 6. And right here, you have the 2, 3, 4, 5, and 6. Of course, you remember that this is upside down. That's how the steric chemistry is working. So you just got this idea that how these are the positions of these different hydrogen bonding positions. And what we are going to see, that you can see the hydrogen bondings are mainly forming in between these amine groups. They are predominant amine groups, and also you have imine groups, as well as you have these keto groups. And what we are going to see in a second, that these forms of these bases actually have a problem which we call as a tautomerization, because they can be found in a different kind of form and they can change, although these are the predominant forms, but they can change to these tautomers, and these tautomeric isomers can actually bond in a different way. And that's why we just have to have an understanding of the structure of the nucleotides. So let's go back to the mutations. So first, we are going to talk about these heritable changes in the DNA. And a point mutation is an alteration of a single nucleotide that we just talked about. And it could be an addition or deletion of one nucleotide pairs. And these are actually the most common. Whereas an insertion or deletions, these are a little bit uncommon, where you either insert a base or delete a base. On the other hand, you have an inversions and the duplications and translocation of a nucleotide sequence can happen as well. So in those cases, you can have the inversion of a sequence, or you can, so say for example, you have an AT and it becomes TA. So that will be an inversion. You can have a duplication of the sequence. Suppose you have an AT and then you just have two of these ATs, AT and AT. That will be a duplication. And then you can have a translocation of the nucleotide sequence, where one sequence can be translocated and somewhere else. And these are much more drastic mechanisms. And that can also happen in the DNA. And there are some of the mechanisms that we are going to talk much later in the chapters as well. And the mutations can be spontaneous, as well as they can be induced by environmental factors. We call them as the mutagens. So the first, spontaneous mutations may result from errors in the DNA replication, and these are head-on collisions between the replisome and the RNA polymerase, where just basically one enzyme knocks off each other and it just makes a mistake. And there are spontaneously occurring lesions on the DNA, and how these lesions are formed. One of the most common way the lesions can form in the DNA, because in the aerobic conditions, constantly in the cell, these free radicals are actually formed, and the O2 minus unpaired electron, which is called the reactive oxygen species, or ROS, or sometimes we call it as superoxides. And these superoxide or the reactive oxygen species, which are also called the free radicals, these can actually cause lesions in the DNA, which can basically sometimes leave this sugar and the phosphate backbone, but they can actually cut out these bases. And we call, depending upon what kind of bases are being cut out, we call them as a pyrimidines, or a pyrimidine actually goes out, or a purine can go out, but the backbone remains the same. Depending upon that, we call that a purinic site, or a pyrimidine site is actually produced. So these are usually called the lesions that can happen. And also, as I said, that the action of mobile genetic elements, those can actually insert, delete, invert, duplicate, and translocate many of the nucleotide sequences. And these transposable or mobile genetic elements are going to cause that. And we are going to talk about those mechanisms later in the chapter, in the part 2. So let's talk about the different mechanisms. The first one is the tautomerization, and the nitrogenous base of the nucleotide shift to tautomeric form and allowing for unique base pairing to occur. Now let's take a look at this, that this is the amine form of the cytosine. And if you just look at that this amine form of the cytosine do contain an NH2 right here. And this is the normal form. And if this is present, this A will base pair easily with a T, or with the cytosine would base pair with a G, basically a guanine right here. So on the other hand, what happens that sometimes this NH is converted into the imine form NH right here, in the amine form to tautomeric imine form. At that time, you can easily see that this amine form will base pair with a amine form of the adenine. So right here, you have an NH2. This is the amine form of adenine. So this is a normal amine form. This is more common, that can base pair with a wrong form of cytosine, which is in the tautomeric imine form. On the other hand, see that this is a normal cytosine, which is in the amine form. This tautomeric form is prevalent, but it can base pair with an adenine, which is in the imine form. So here, this is the tautomer which can cause this problem. This is the tautomer which can cause this problem. This amine form of cytosine and amine form of adenine, these are the normal base pairs. But when the cytosine or adenine, they are with the wrong tautomers, they can form this. On the other hand, see that right here, in between the thymine and the guanine, there are two different tautomeric form, which is a keto and enol form. They can form this. So let's take a look at this thymine right here. So here you can see the C double bond O right here. This is a keto form of thymine. Whereas see that right here, C double bond O, this is a keto form of the guanine. And these keto forms are actually the normal forms, which actually do properly base pair. And this guanine will base pair with a cytosine in a proper way, and this thymine will base pair with an adenine in a proper way. But what happens that when a guanine actually changes from its keto form to enol form, because see that this is like a CHO, this is the enol form, so you have an alkene and then you have an OH, all, which is an alcohol, alkene, alcohol, enol. So this is an enol form, and when the enol form actually forms, which is not the desired form of the tautomer, that can bond with the keto form of the thymine. On the other hand, see that what happens that when there is an enol form of the thymine, the normal, this is a keto form, when enol form of thymine, this is a wrong form of the tautomer, this can actually bond with a normal or a keto form of guanine. So here you can see that instead of a G and C, you have a G and T base pair. Instead of a GC, you have a GT base pair. And this is due to the tautomerization of either one of them. On the other hand, you can see that these are the CA base pairs, and this can happen to the amine tautomerization of each one of these, one of the bases. So this is the tautomeric shift that can cause these unfavorable base pairing. The next one is the transition and the transversion. The transition basically lead to a stable alteration of the nucleotide sequence. On the other hand, transversion is a little bit less common, where a purine is substituted for a pyrimidine, but this can cause a steric problem. But that is the reason that it is a little bit uncommon, but it is still observed. But transition is much more common. So here is what we are looking at, that what happened to this parental DNA, and as a transition, as you can easily see right here, the tautomerization is the cause for that. So here you can see that this is a normal GC base pairing. But in the normal GC pairing, this is temporary enol tautomeric form. Here you have, instead of a GC, you have a GT base pairing, which is basically a GT base pairing. This is due to the tautomeric form of the guanine. So in this case, you have see that this is the parental DNA, and now they are being replicated. So what is happening that in the first generation progeny, you can see that this strand was a parental DNA with a CG. This is perfectly fine. This is a wrong base pairing. So the first generation progeny, it gets a CG. So again, the second generation, there is no problem with there is no mutation, and both of them are the wild type, means they are the same as the parental DNA. But what happens that in this DNA strand, you have these tautomeric shift, and that causes a GT base pairing. And now in the first progeny, now when this DNA is replicated, the two strand will separate. So this will become a G. This remains as a G. So a C proper base pairing will happen, and this will become a wild type. But the problem is what happens that due to the temporary enol tautomeric form of the guanine, this thymine wrongfully base paired with it instead of a C. So here what you have in this strand, you have a T, and an A will base pair with it correctly. And now you have a mutant form, because this is this is not the original nucleotide. So here you can see the original nucleotide was TGCAG. Here you have a TGTAG. But on the other hand, the other strand with this TGCAG, this is perfectly fine. These are two perfectly fine as well. So this is an example that when there is a tautomeric shift and unnatural base pairing, that can lead to a mutation which can be carried forward as well. All right, so after that, we have some more drastic effect, which are the insertion and deletion, and occur at short stretches of repeated nucleotides. And in this process, the pairing of the template and the new strand can be displaced. So what happens that here, what we are looking at a sequence where you have these multiple A's, A. And when DNA polymerase is actually synthesizing this, sometimes there is a condition which we tell is that DNA polymerase actually slips due to this. And when this slippage actually occurs, it basically forms a kind of displaced new strands like this, basically, and then it will keep going on synthesizing the rest of the strand. And in that process, see that this T was essentially, so you have 1, 2, 3, 4. 4 T's were synthesized, but as it is slipped, this is basically T. These are bonded. This T is now as a loop staying outside, and now an extra T will be added right here. So an extra T is added right here. So because this T was originally in there, and now you have an insertion. And this will be the DNA strand essentially. And as soon as you have this insertion, what happens that just due to the insertion, the entire reading frame of a protein coding gene or RNA tRNA coding gene or the ribosomal RNA gene coding gene will be out of frame, or you will read the sentence of the codons in a different way. And the same way it can happen with the slippage with the deletion. So here the slippage is happening in the parental strand. Here the slippage is in the new strand. Here the slippage is in the parental strand. So here, when this is happening, you can see right here, you have these continuous TTs and then GC, and then it slips out, and then you have an A. And at that point, this A is basically, you can easily see that when this essentially happens, you have initially you have an GCA, A, and that's why you have a CG and then 4 T's. Now you have this, and you have this CG, and then you have this T, and T's. And what happens that just due to this, this base of this A, one of the A, initially it was 1, 2, 3, 4, and 5 A's. This is deleted. You have 1, 2, 3, 4, 4 A's basically. So, and right here, you have a deletion. So originally you have 5 A's, 1 A is deleted. Now you have 4 A's. So the same way that the reading frame of the gene will be completely changed. And due to this, the insertion of deletion of the base pairs of the bases will cause very drastic mutations, which we often call as a frame shift mutation. So after that, we are looking at the lesions in the DNA, and these actually form what we call as an epurinic site, which cannot base pair and may cause mutation in the replication round. So the same way that as an epurinic site can be formed, depyrimidation of cytosine and thymine residues can also occur, but at much slower rate than the depurination. So what happens in the depurination, you can see a guanine actually goes out and an epurinic site is formed. Remember that this happens spontaneously due to different lesions that like a reactive oxygen species and something, and many free radicals and other environmental factors. And in this DNA, remember that this is not a deletion. Remember that in the deletion, the entire nucleotide is deleted from here. The phosphodiester bond is broken and deleted. Here it is not that. It is only the base is deleted and the phosphate backbone as well as the sugars remain in place. And that's the reason that we actually talked about that. So basically what happens, the phosphate backbone and the phosphate and the sugar remains in place, just only right here in the base there will either adenine or guanine as a purine, and this glycosidic bond is broken, and this is what we call as a lesion. So just only the base is removed. So once this base is removed, you can see that when the strands are separated for the first round of DNA replication, there's no problem with this strand. You have an ACCG, TGGC, and there's no problem with that. So this is wild type, nothing happens. But now what happens right here, that without a proper template, an incorrect nucleotide is basically incorporated in during the DNA replication. So it just randomly puts an A and CG right here. And because there is nothing to base pair with. So here you have just a phosphate and you have the sugar, but there is no base, there is no G in here. So as there is nothing, it just randomly puts an A here, and that is carried forward. Now what happens now in the new strand, in the second strand separation in the next generation, you have an AACG. Originally you have in this an ACCG, now you have an AACG. And in the second round of replication, a complementary T will there. So this will be a mutant form, and this will be carried forward and using generating a mutation, and this will be carried forward. On the other hand, you can see that this strand, which is still with the epirenic site, this can actually put another different nucleotide in this place. It doesn't have to be an A. Here first randomly it put an A. Here it put randomly another G right here. And now it will be an C. So initially it was TGGC. This can in the next round can be TCGC. And instead of ACCG, this could be an AGCG. So if in this strand, will basically create an a site which will cause the second route of DNA replication and will carry forward with the incorrect nucleotide. So these are basically the lesions in the DNA which can cause the mutations. So after that, we are looking at the B, which is the induced mutations by the mutagens. So the number one that what we talked about is the spontaneous mutations. And we talked about tautomerization, transition, transversion, and insertion deletions and lesions in the DNA. Now we are looking at the induced mutations, and there are several different type of mutagen that can basically physical chemical agents that can damage DNA. One of the classes of they are base analogs. They are structurally similar to bases, and they can change their tautomeric forms very rapidly, much more than a normal base, and they can be incorporated into the growing nucleotide chain, causing a mismatch. And there are DNA modifying agents, and these can alter a base, causing it to mispair. And also there are some agents which are called intercalating agents, which distort the DNA and induce a single nucleotide pair insertion and deletions. So let's take a look at these, a couple of them. So here, these like 5-bromouracil or say 2-aminopurine, these are the are the base analogs. On the other hand, you can see this methyl nitrosoguanidine, ethyl methyl sulfonate. These are alkylating agents. So basically, they add a methyl group to the guanine. Alkylating agents basically are a class of compound which can add a methyl or an ethyl group. So this ethyl methyl sulfonate, on the other hand, adds an ethyl group to the guanine. And then hydroxylamine, these are all DNA modifying agent, hydroxylate cytosine. Nitrogen mustard, alkylating agent, adds a methyl group to guanine. This is the nerve gas, mustard gas. Nitric oxide deaminates bases, and then ethidium bromide, actinium orange, these are called intercalating agents. And then we will see that the physical mutagens, which are UV or X-rays, can also promote pyrimidine dimer formation, particularly the thymine dimer formation. And X-rays can also cause base deletion, single-strand nicks, cross-linking, and chromosomal breaks. So let's take a look at this, how these basically work. So the first one is the base analog, and we are taking a look at the bromouracil. So this is very similar, a base analog, similar to thymine. You can see that there is a bromine instead of a ethyl group right here. and they can be in the keto form. Right here you can see the keto form, whereas this hydroxyl right here is the enol form. So you can see the 5-bromouracil in the keto form base pairs with an adenine right here, and a 5-bromouracil in the enol form can actually base pair with the guanine. So essentially what happens, the problem is that as the base analogs actually can appear in their tautomeric form much more frequently than a regular base, they can actually cause this mismatch. So this is 5-bromouracil in action, causing mutations. On the other hand, the chemical, which are DNA modifying agents, there are different ones. They change a base's structure and alter its base pairing specificity. So DNA alkylating agents are among the most consistently identified class factors. They cause DNA damage and the mutations. So what they do, that they basically add alkyl groups to specific bases and resulting in alkylation products. So basically the O2, so these numbers basically are the carbon positions, which basically we saw in the very beginning. So if you just referring to these numbers, just refer to these numbers basically. So you will understand that where this is actually happening. Although details of that not necessary, but see that this is the oxygen of alkyl thymine. This alkyl group is present in this second position. Alkyl thymine, fourth position. Methyl guanine, sixth position. Ethyl guanine at the sixth position. So basically in this 2, 4, 6, and 6, these are the positions where these oxygens are basically methylated or ethylated by the alkylation. And it could be a monoalkylation, it could be a cross-link in between the strands, and it could be an intrastrand cross-linking, and this is also the intercalation of different products that can happen. And this, all of these modifying agents will basically distort the DNA and their base pairing ability. So here is an example of methyl nitrosoguanidine, and it is an alkylating agent. So this adds a methyl group, the O6 of the guanine. So here you can see that this is a guanine, and these are the 3 hydrogen bonds. This is the amino form of the guanine. And right here is this N-methyl N-nitrosoguanidine, and commonly this is just known as the nitrosoguanidine. And this base right here, this methyl base, is basically put onto the O6 position. So this is like a 1, 2, 3, 4, 5, and 6. This is 7, 8, 9. So 6th position O is now methylated. So this is what we talked about right here. Methyl nitrosoguanidine, alkylating agent to the group guanine, and see that this is an O6 methyl guanine can be formed as an alkylation product. So this is O6 methyl guanosine, and sometimes this will pair with the thymine because this will not pair anymore. Hydrogen bond cannot form with this alkylating agent, which with this methyl group, you cannot form a hydrogen bond. You can only hydrogen bond in these two positions, which is 1 and 2. The 6th position is not available for hydrogen bonding, so this will not bond naturally with the cytosine. So this basically mispair with the thymine. So this is the nitrosoguanidine's effect as a DNA modifying and alkylating agent. Then is we're talking about the chemical. These are called the intercalating reagents, and they themselves put themselves between the base pairs. And intercalating agents often are fluorescence, utilize the DNA analysis and the microscopy. And of course, these are mutagens as well. So here is an example which is called the ethidium bromide. And this ethidium bromide, this is the planar compound, and they can intercalate themselves in between the DNA. So the ethidium bromide, on the other hand, of course, is a mutagen, but it is widely used in the staining the DNA in a DNA gel. So what you are seeing in here, that this is an agarose gel where each of the lanes are loaded with the DNA, and what you are seeing that these are the different DNA bands. And after the gel is run, or during the run, you just expose them to ethidium bromide. There are two ways. Either you can add the ethidium bromide when you are loading the gel, or you can just put them in an ethidium bromide containing buffer for a little while, so that the DNA actually soaks up the ethidium bromide and intercalates themselves, whatever you prefer. And after that, this is called an UV, ultraviolet trans illuminator, and you have to take ultraviolet protection. It can completely cover your face, which is UV cannot penetrate that acrylic, special type of acrylic, which is UV resistant, and you have to have a clothing for that as well. And when you actually put your gel in on top of that, you will be able to see this orange glow, which is the fluorescence that is coming out from the intercalating reagent, which is ethidium bromide. Now, there are many of these, like say, for example, acridine orange, proflavin. These are usually used as an intercalating agent as well. And acridine orange, these are used in fluorescence microscopy as well. But there are many compounds like these which are intercalating agent, and they are mutagens as well. So, if they're occurring naturally, they can mutate the DNA in the environment as well. So after that, we are looking at the induced mutation by a physical mean, and this is basically what we are looking at, the physical mean, and the example with is an ultraviolet light. In the presence of ultraviolet light, there is a very unique reaction happens, a thymine dimer, and this is a thymine dicyclic thymine dimer that can be formed right here. So the thymine is basically base pairing with, so T, these are two T's. So on the other side, there should be the base pairing with adenine with these two. So instead of base pairing with an adenine, those hydrogen bonds are broken, and they actually form this thymine dimer on one side of the DNA. This is just only one chain area you're seeing. This is the sugar phosphate backbone. So this is a thymine dimer formation. And this will basically cause the DNA polymerase to stop at that point of time and cause different kinds of mutation. But we will see that there is a process by which these dimer can actually be removed by a repair mechanism or process. So after that, we are looking at the effects of the mutation and how the mutation is carried forward. So we already talked about that. What is the wild type, most prevalent form of the gene and associated phenotype? And the forward mutation basically changes the wild type mutation, changes the wild type to a mutant form, which is less common and is different from the wild type. So we are looking at that when we are looking at a protein coding gene, the point mutation in protein coding genes can affect the protein structure in a variety of ways. And they are called a silent mutation, missense mutation, a nonsense mutation. And usually the silent, missense, or the nonsense mutations will cause due to change in a base, basically. On the other hand, what happens that the frame shift mutation actually can cause from an insertion or a deletion, basically. So let's take a look at each one of them. These are the definitions and each one of them in action. So here what you are seeing that this is a wild type and this is a sequence, and the silent mutation in a substitution, this C is substituted by an A. But what happens that this is the third position of the triplet, which will make the third position of the codon different. But due to the degeneracy that we talked about, that many of the amino acids are coded by multiple codons, and ACC and ACA, which is a change in the third position, is often code for the same amino acid, and in both cases a threonine is formed. So what happened that in a genetic level or genotypically, these two genes are different with a different base in this place, but phenotypically the protein is going to be exactly the same. So there will be no effect on the there will be no effect of mutation, and we call this as a silent mutation. Now what happens is a missense mutation. In a missense mutation, you can see you have a TCC, which codes for a serine. Now this C is basically substituted by a G, and now it codes for a cysteine. This is a different amino acid instead of a serine, and this we call as a missense mutation. Sometimes if you have just a mutation at a site which is not very important for its three-dimensional structure or catalysis, this is well tolerated. But if it changes some of the critical nature of the proteins, the protein can prove dysfunctional, or if it is an enzyme, may prove that it is less effective as well. So these things can happen. On the other hand, a nonsense mutation is also by a substitution. And see right here, you have an AAA, which codes for a lysine. And this A is substituted by a T and TAA, which will form an UAA, that is a stop codon. So your stop codons are UAA, UGA, and UAG. So this is UAA, will be a stop codon from this triplet. And the protein will basically stop. And if this stop codon is pretty much in the beginning or sometimes in the middle part of the protein, almost always this will cause the protein to essentially not functional. The protein is basically dead, and the organism will not have that protein. If that protein is necessary for its survival, it will be a lethal mutation. But if this stop codon, premature stop codon, basically is towards the end of the protein, where just a few base pairs away from the end, and that may not cause a drastic effect of the protein. It may be a little bit less functional, but it may not be lethal. But it just all depends what protein, where these premature stop codons can be inserted. So nonsense basically mutations are basically when amino acid is replaced by a stop codon. Then we are looking at the frame shift mutation by an insertion or deletion. So we already talked about that, how the insertion deletions actually can happen. And you can easily see that here you have 4 C's, C, 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 C. So we talked about that when you have these kind of sequences, sometimes the DNA polymerases can actually slip on these. And this is exactly what happened. And a G is inserted in this. So up to this is perfectly fine, methionine, threonine, serine. As soon as you put a G in here, so the entire reading frame after this is changed. So instead of CCG, proline, AAA, lysine, and GG, glycine, now this G and the reading frame will change to GCC, GAA, and AGG. And this will change to alanine, glutamic acid, and arginine. So the entire reading frame is now changed. And usually these will lead to very drastic mutation. And if this is most of the time, if this protein is required for such function, the insertion or frame shift mutation will render the protein non-functional, unless it is in a part which is towards the end of the protein where it may get away with this. But most of the times, this protein will not be functional due to these frame shift mutation, which is very drastic. On the other hand, deletion, the same thing. Here also the DNA polymerase can slip, and one base is removed in this process. And as this is removed, you have, instead of 4 C's that we have C, 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 one base is removed. Now you have 3 C's. And after this, you have a CGA, which is an arginine, and AAG, you have a lysine. And here also the reading frame will be changed, and the effects will be the same as an insertion or the deletion. Most of the time, the protein will be non-functional in these conditions. So after that, we are looking at that once these mutations happen, there could be also reversion of the mutation, which can revert a mutant phenotype to a wild phenotype, or there could be a suppressor mutation, or where the wild phenotype is restored by a second mutation at a different site than an original mutation. Now let's take a look at that, how the reversion and suppression of mutations happen in action. So here we are looking at a sequence where you have this same methionine, threonine, serine, proline, lysine, glycine, and you have A substituted by a G, and now it is an alanine instead of a threonine. So what will happen to this protein? This is just only one amino acid, but a threonine is basically a polar amino acid and is substituted by a non-polar amino acid. And if this is involved in some kind of folding of the protein, just the substitution of the alanine basically can make the folding impossible, and it can basically make the protein non-functional. On the other hand, see that the G is now again reverted back to A, and it forms the threonine, which is the original wild type. So right here, this is a straight reversion back to its original. So this can happen as well. So after that, we are looking at that this is basically what is called, so this is a reversion to restore the original mutation. The second type is reversion and intragenic suppression by a base substitution. So what happens here? You have this sequence, and the first, it is substituted by G, and now you have an alanine. Then you have a second mutation where this G is substituted by a T, and now this ATG, after that the TCC, is basically a serine. So what is the difference between this and this? Here you have a threonine, which was converted into the alanine. This is a non-polar amino acid to a non-polar amino acid. So this is basically a drastic change, and sometimes that can cause many problems with the DNA, for many problems in the protein for its function. But now it reverted back to serine. Serine is a polar amino acid, and due to that this serine can be well tolerated by the protein most of the time, and sometimes not, but most of the times, if you change a polar amino acid to a polar amino acid, or a non-polar to a non-polar, or a hydrophobic to hydrophobic, or a hydrophilic amino acid to a hydrophilic amino acid, and that those are basically well tolerated. And we call this as a reversion and intragenic suppression. So basically a change in the DNA as well as in the protein, but it may not have a drastic effect, and this mutation can be carried forward. So after that, we are looking at the reversion and intragenic substitution by a frame shift generated by a one base insertion. And let's take a look at this. And then often insertion of a single amino acid is well tolerated by the protein, but sometimes it may vary. And then what happens that the one base is insertion, the reading frame is then again restored by a one base deletion. So let's take a look at how this basically happened, which is a reversion and intragenic suppression as well. So here, this is the sequence of the protein, and you have right here, you have a G. This is an insertion, one base insertion. So the reading frame completely changes. So here, see, from proline, lysine to glycine, this is an alanine, glutamic acid, and arginine. So this could be very detrimental to the protein, and probably it will be non-functional. Now what happens? There is another base, which was a T. This T is basically deleted from here. So this T is deleted. So you have a CC, then you have a CC. This T is deleted. So in this process, what happened? That you have, see that right here, ATG and ACC. This is a threonine, and now you have an extra proline right here, which was earlier serine. So you have this proline, lysine, and the glycine. The rest of the protein is the same, just only instead of a serine, you have a proline in this place. So this is a different amino acid than the serine, but the rest of the amino acid are the same, and sometimes this could be well tolerated. Insertion of a single amino acid is sometimes well tolerated, but sometimes it may change function, but most of the time this can be tolerated. So this is basically a reversion and intragenic substitution, which by a base insertion followed by a deletion. On the other hand, there could be extragenic suppression, is basically a nonsense suppressor forms elsewhere in the chromosome that can actually happen. And sometimes a tRNA anticodon, if allowed to base pair with a nonsense codon, which may basically rescue a gene, basically. So what we mean by this, that suppose there is a condition that a premature stop codon, TAA, is put into a gene, and the product of that gene, which is the protein, is non-functional. Now what happened? That we have a problem with a tRNA where then anticodon is now base pair with a nonsense codon. So now this tRNA basically will put, see right here, there was a lysine, and the TAA now basically puts a lysine in that place. So this is basically a mutation in the lysine codon, which can have a drastic effect, of course, as well. but this may rescue one gene, but it may have problems in other places by putting different tRNAs. But the thing is that what happens, that remember that each of these amino acids can be coded by multiple tRNAs. So there is a degeneracy. So there would be other lysyl tRNAs which can function and make correct proteins. But just only one lysyl tRNA, the TAA, can bring, instead of a stop codon, making it to stop, it may bring the lysine and can rescue the gene, basically. So that's how we call this as an extragenic suppression, where a T anticodon can allow to base pair with a nonsense codon to rescue a gene. So after that, we are looking at some other type of mutation, which are called conditional mutations, different phenotype in an environmental condition. And sometimes we call, some of these are called the auxotrophic mutant. And these auxotrophic mutants are basically lethal. They are unable to make an essential micromolecule such as an amino acid or nucleotide. So these are the conditional phenotypes. So what happens that when the auxotrophs are supplemented with that macromolecule like the amino acid or nucleotide, then they can survive. Otherwise, in a normal condition, if you do not supply it with what they are missing, that would be a lethal mutation. And the wild type strain from which they arose is known as a prototroph. Auxotroph is deficient in something. Prototroph is the original, which was not deficient into that. So the other type of can happen is the mutations in the regulatory sequence, and these can control gene expression. Mutations map in the operator produce altered sequences not recognized by the lac repressor protein. Operon can be continuously transcribing beta-galactosidase, is always synthesized. So basically, inducible genes are not inducible anymore. They become a constitutive gene, and they can basically take out all the energies from the cell. And these, even if there is no lactose in the environment for an E. coli, just for saying, it is still making all the beta-galactosides, permeases, trans-acetylases all the time, because there is a problem with the lac repressor protein. It cannot bind to the operator to stop the gene expression of the lac repressor. And also we talked about the tRNA and ribosomal RNA gene, which protein synthesis can be disrupted. Although there are very interesting observations, and we'll talk about that somewhere else, sometimes these ribosomal RNA genes can alter the ribosome, and without the altering the function of the ribosome. But what happens that there are particularly some drug targets, which basically, like erythromycin, azithromycin, these basically target the ribosome. And sometimes these ribosomal RNA genes make the changes in the ribosome in such a way that these antibiotics are no longer can bind to the site, and these strains become antibiotic resistance. So these things can also happen due to the mutations. So these are mutations in the tRNA. Ribosomal RNAs are low frequency of mutation compared to protein coding genes, and usually lethal because their 3D structures are critical, and the mutations may impact translation. But again, out of many mutations, it just only take one mutations to make them fight antibiotic resistance with that, and that can happen as well. And these mutations are initially identified based on the slow growth as well, because what can happen that there are multiple tRNAs for just one amino acid. And if one is mutated, sometimes you can see the slow growth, as we talked about that. So after that, in the detection and isolation of mutation, and we are going to talk that how we can get an auxotroph and how we isolate and basically understand that what they are going on. So usually the mutation rates are in between 1 per 10 to the power 7 or 10 to the power 11 cells, and finding mutants requires a very, very specific sensitive detection method. Because what happens that many a times a particular auxotroph may not show any kind of physical differences. These colonies are cream color. The auxotroph and the prototroph both may be the same. If they have a blue or red colonies, say a mutants or a change in color, then it's very easy to identify. But if not, we use this replica plating technique to screen the auxotroph. So how we do this? So here we have an E. coli with a mutagen, say for example like a methyl nitrosoguanidine, and then we just basically allow them to grow and we put them on a master plate. And this is a complete media. That means it contains all the amino acids, and even the auxotrophs will grow and the prototrophs all will grow. And here is what is called a replica plate, and this is a replica block, and here is a velvet sterilized, and we touch this replica block onto this master plate, and then we have these replica plates. Few of these replica plates will have a complete media with all the amino acids, and some of these replica plate will have a medium. Say for example, we are trying to find a lysine auxotroph. So these replica plates will not have lysine, and we touch the replica plates in each places. See that right here, it is shown, the auxotroph is shown as a red color, but we may not have that red color. This is just for the identification. So here we have the all stains are growing in here, and here the auxotroph will not grow. So as these are replica plates, we are going to watch that whether you have at the same position the colonies. We are going to compare. You have a colony here, we should have a colony here. Of course, they will be aligned in a proper way. And we have a colony here, we have a colony here. But what we will see that we have a colony here, but that colony is missing. So we may take this colony from the stains grow, because here this is a complete media, all the strains are growing here. We can take this one and culture this and try to do the next round of experiment to grow them in a complete media and without lysine. And if that isolate doesn't grow in the lysine, you have isolated a lysine auxotroph. So these are some of the things, and sometimes the addition of mutagens can revert an auxotroph to prototrophy as well. So after that, we are looking at the section DNA repair maintains genome stability, and this is actually a very, very important chapter. And even the 2015 Nobel Prize was actually given to three scientists who actually elucidated the mechanism of this DNA repair, and tremendously important. And from the repair mechanism of the prokaryotes, we actually that actually led our way to understand the DNA repair mechanism in the higher eukaryotes as well. So let's take a look at some other information. Is that the SARS-CoV-2 mutation is nothing to do with this chapter directly, because we are talking about the bacterial mutation by the DNA polymerases, whereas the SARS-CoV-2 mutations, this is due to the faulty RNA polymerase, which the SARS-CoV-2 virus does have by itself, because it's a plus-stranded RNA virus. It codes for its RNA polymerase. But interestingly, that RNA polymerase is the only RNA polymerase which does have the proofreading capability in the RNA virus world. In the RNA virus world, coronaviruses are the largest of the RNA viruses, and only the coronaviruses do have these RNA polymerases, which are RNA-dependent RNA polymerases with the proofreading ability. But even with their proofreading ability, that we are seeing so many different variants. And the only reason is that so much of the population was infected. Remember that these RNA polymerase is making a mistake in every replication cycle, every cell that is infected in a single human being. And when the millions of these humans are basically infected, just think about how many cells are replicating these viruses and how many possibilities of mutations can happen. Even with the proofreading ability, their replication is explosive and providing ample opportunity for mutations. And if you just like to go to understand that right from the beginning of the pandemic, how even today that the genetic epidemiology of the SARS global variants, you will be able to just basically for this nexttrain.org, you can just follow this link and you can see the animation of the entire mutations. And those mutations are mapped to the entire 30 kilobase genome of the SARS-CoV-2 genome. So you can see that where these mutations actually happen and how they are selected, and one after another, how the dominant strain from alpha, beta to from delta to omicron, how over the period of time there was waves of different dominance that actually changed over the period of time. And these are due to just the selection pressure that the viruses doesn't actually think. It just basically the environment selects the variants based on its ability to basically propagate. So essentially what happened initially, there were some of the deadly strains, and we are now seeing that the strains are not deadly that much, but they are more infectious because many people are vaccinated. We are using masks and other protective measures. So the chances of infecting a person is less. And that means that a virus, which will be a lot more infectious, has the advantage of the viruses which are less infectious, and those get selected over the period of time. So this is just a thought of mutations that in action in a different system, which is the viral evolution. How mutations affect our lives as well in a pandemic or any kind of like these disease. So after that, we are talking about the DNA repair, and there are two parts of it. The first is that what we are not going to talk about is the proofreading. So this proofreading is an inbuilt mechanism of correction of errors, base pairing during the DNA replication, and they are done by the DNA polymerase 1, DNA polymerase 2, and the DNA polymerase 3. So DNA polymerase 3, as you know, is the main replicating enzyme. DNA polymerase 2 is a backup of the DNA polymerase 2, and it can function at a low ability to replicate the DNA, chromosomal DNA as well. And DNA polymerase 1 is basically filling the gap and repair function as well. All of these DNA polymerases have proofreading ability, and they just kind of scans and scoots back and forth, excise wrong DNAs, and fill those gaps as well. So very interesting in this process as well. So very interestingly, what happens, the DNA polymerase 1 was discovered by Arthur Kornberg, and that was in 1959, a Nobel Prize by the Arthur Kornberg. And then that was the first. The DNA polymerase 2 and 3, 4, 5 we have in the DNA system. Those were not given Nobel Prize. The first one was given. On the other hand, the same way, this is just a little bit out of the way, but the 2006 Nobel Prize was given to Roger Kornberg, who is the son of Arthur Kornberg, who actually discovered the RNA polymerase 2, which is the main mRNA making enzyme in the eukaryotes. So many discoveries. On the other hand, very interestingly, he did not get the Nobel Prize. The DNA polymerase 3 was discovered by Thomas Kornberg, and Thomas Kornberg is the brother of Roger Kornberg, son of Arthur Kornberg. He did not get the Nobel Prize. So in the same family, Arthur Kornberg, dad, got the Nobel Prize for DNA polymerase 1, Roger Kornberg for RNA polymerase 2, also Nobel laureate, and his brother Thomas Kornberg discovered DNA polymerase 3. So very interesting family of the Kornberg families for the discoveries of these DNA RNA polymerases. But the interesting part is that the 215 Nobel Prize in chemistry was given to these three people. Thomas Lendl from Sweden, then Paul Modric is a Croatian American, and then Aziz Sankar, he is a Turkish American scientist, and they basically understood the different type of these repairs. So we are going to basically talk about their methyl-directed mismatch repair, which was pioneered by Paul Modric, and the nucleotide excision repair by Aziz Sankar, and base excision repair by Thomas Lendl, and immensely important for our understanding of how the genome stability is basically maintained. And then we're going to talk about the direct repair by photoreactivation and alkyl group removal from guanine, and a recombination required mechanism. So DNA repair is an extensive interest because this is basically not only in the prokaryotes or the bacterial archaeal world, but this is also extrapolated in the eukaryotic world. So, by which the sanity of the DNA, or the so complex sanity of the DNA, is basically maintained, and the effect of the mutations are being fought back constantly. But still, we know that we have mutations, and due to that, we have all the different genetic disease. But without these mechanisms, it would have been much, much more drastic. So the first one is the mismatch repair, and this is what we called as a methylation-directed repair. So what happens that we will talk a little bit extra compared to the textbook. So in this process, this mut SL and H complex basically helps in this process. So these are three proteins essentially. So the mut S basically scans the DNA, and it binds that close to where there is a mismatch. And then with mut S, mut L is also recruited. On the other hand, the mut H actually finds that where there is a DNA methylation. So what is the DNA methylation? So the DNAs at a certain sequences like GATC, these adenines are methylated, and this is basically a mechanism by which the nucleases cannot easily digest the DNA, and this is basically for the stability of the DNA. So that's why the DNAs are methylated. So what happens that when a newly formed DNA, so during the replication, when a new DNA strand is synthesized, it takes a couple of minutes before the newly synthesized strand, which is the purple strand is the newly synthesized strand. It takes a little time for it to get the methylation. Whereas the parental strand, which is the methylated strand, that is already methylated, and so the strand just after the replication, we call this hemimethylated. That means that hemimethylated means the parental strand is methylated and the daughter strand is not. And this replication mechanism need to happen within that window. So now the recognition is done by the mut H, where these methylations are, and then this mut L and UVRD basically separate the two strands, and it nicks out where the closest these methylations are. So the only part which is the daughter strand in between the two methylation, that is removed. So the purple strand, which is the daughter strand, is basically removed, and the parental strand with the methylation is stabilized by single-stranded binding DNA, single-strand binding DNA protein molecules, basically. So now what happens that the same way the repair mechanism is the same, that DNA polymerase 1 basically synthesize these gap basically based on this parental strand, which doesn't have the mutation. The mutation was only in the newly synthesized strand, and now the gap is basically sealed by the DNA ligase that we have basically seen. So this is basically the methylation directed repair. So it cuts the DNA if there is a mismatch in between the methylation, and the basic idea is that the newly synthesized strand has a mutation, and that newly synthesized strand is removed, and the parental strand is kept, and the mutation and the DNA repair mechanism by DNA polymerase 1 basically get the original strand back without the mismatch. And this was the work of Paul Modrich. And after that, we are looking at an excision repair, and this corrects the DNA that distorts the DNA double helix. And there are two types. One is a nucleotide excision repair and a base excision repair. And it removes the damaged DNA and uses the intact strand as a template to synthesize the new DNA. So the first one is a nucleotide excision repair and a work of Aziz Sankar. It removes thymine dimer, and it involves a system which is called an UVR ABC endonuclease system. So the UVR endonuclease system basically scans these, and sometimes a thymine dimer can be found, and it will basically bind into that region, basically, because there is a distortion. It could be other distortion as well. So UVRA basically recruits UVRB, and UVRB then recruits the UVRC, which is the endonuclease, and it cuts a few bases on each side of that mismatch, basically. So this is the nucleotide excision. So this is excise out, and DNA polymerase basically fills the gap, and DNA ligase seals the gap. The next mechanism is known by the base excision repair, and this was basically discovered by Thomas Lindell. And this basically removes a damaged or unnatural bases, yielding basically a epiremic or an epiremic site. And where the backbone remains the same, and due to a lesion, just the base is excised out. And if this happens, a class of enzyme known as DNA glycosylases remove these damaged unnatural bases. So as you can see that each of these mechanisms actually involved an entirely different mechanism. One is a mute SLH and UVRD related repair. One was UVR ABC related repair, and one is DNA glycosylase and AP nucleases based repair. All are different systems and discovered by different scientists. All these scientists, and all together they got the 2015 Nobel Prize. So once DNA glycosylase removed the damaged unnatural bases, the AP endonuclease recognized the damaged DNA and nicked the backbone of the AP site. And then the DNA polymerase 1 DNA ligase repaired the site. So here you can see that instead of an AT repair, an uracil is incorporated into the DNA. And DNA glycosylase recognizes an abnormal base and cleaves the bond in between them. See that right here, the phosphodiesterase bond is there, but this is an epiremic site. So that only the uracil, only the base from the first carbon glycosidic bond is basically digested by the DNA glycosylase. Once that is removed, another enzyme comes in, AP endonuclease, recognizes a gap, and now it cleaves the DNA backbone on the 5' side of the missing base right here. So now what happens? Now the same way that DNA polymerase 1 now uses its 5', 3' exonuclease activity to remove the damaged region, and then it fills it with a normal DNA, which is correct, and then DNA ligase basically seals the gap. And this is your base excision repair. So all of them achieve the same thing, but in different way by different mechanism. Then there are some direct repairs, which is called the photoreactivation. And this photoreactivation is done by an enzyme called photolyase. Now what happens, under the ultraviolet, the thymine dimers actually can form. So we can see that the thymine dimers can be removed by this process as well, nucleotide excision repair. There is another repair mechanism, which is the photoreactivation. And this is by the enzyme called a photolyase. So if a thymine dimer is formed during the ultraviolet exposure, when it becomes dark, this photolyase basically scans the DNA and look for the thymine dimers, and it binds to the thymine dimers in the dark. After that, if the bacteria is exposed to the light, like daylight, not ultraviolet, these thymines are then separated by a photochemical reaction, and this is catalyzed by the light-activated photolyase. So very interesting enzyme, and why we call this a photoreactivation, because the DNA was damaged due to the ultraviolet, and in the dark, the enzyme responsible for removing the dimer actually finds where the damage is. And when it gets back to the light activating that enzyme to remove that thymine dimers and form the correct DNA. And also there are some enzymes which are like these methylguanine methyltransferases, which can directly repair methylated or alkylated bases. So this, for example, this methylguanine methyltransferase can remove an alkyl group from the O6 position of the guanine. And this is just one example. There are other groups of enzymes which are the direct repair of these methylated or alkylated bases, can be removed from the DNA as well. So the last one is the recombination repair, and this corrects the DNA both bases of a pair missing or damaged. And this involves a recombination with an undamaged molecule. And this can only happen that when there is another copy of the chromosome is available. That means that this is happening during the replication, when the replication forks are separating and there is still another copy of that chromosome is basically present. And this is done by an enzyme, which is a recombinase known as REC-A. So here what we are looking at, that this is the parental strand and the two strands are basically synthesized. One is, this is the newly synthesized strand and newly synthesized strand. And now we have a thymine dimer, and we have a gap in this newly synthesized strand. The reason for this is, you can imagine, if this C strand is basically being synthesized as an Okazaki fragment, where you have a distortion of the DNA, no primer is putting any primer, and that part is basically a gap. So primer, then you have a DNA, then a primer, you have DNA, but you have a gap right here because this is where the DNA is basically distorted. So essentially what the REC-A basically is doing, it cuts out this entire portion of the DNA with this gap, and it takes it out and put it into a homologous portion somewhere else, and cuts this portion of the correct DNA out and swap it right here. So this is basically a swapped region. So you can think about that this is basically almost like an homologous recombination process, basically. So now what is happening, that you have this gapped region right here, and this gapped region can be filled out with the DNA ligase, with the repaired by the DNA polymerase 1, and the gap can be sealed by the DNA ligase. So you have this strand perfectly fine. Now you have a thymine dimer, and this can be repaired by another repair system, which could be very well a nucleotide excision repair right here, and it can be removed by an UVR ABC system, or even it can be removed by the photoreactivation by a photolyase. So this is basically a repair recombination required by swapping portion of the undamaged DNA with the damaged DNA. The next one is the SOS response, and this is an inducible repair system, which is called a global control system, and used when the damage is so great that a normal repair mechanism will not work. This is basically the difference between that the E. coli or any other repair system mechanism is allowing the cell to be dead or make a last try for its survival. So usually when this happens, the DNA is fragmented in such a way that it is beyond basically normal repair. And at that time, basically there is a protein which is REC-A. This is the recombinase protein. And this also does have a protease function, and it is activated. And this LEX-A is a repressor, which basically repress activation of more 50 genes. And the LEX-A is destroyed by the protease. And once the LEX-A repressor is basically destroyed, it activates more over 50 different genes, which is of importance for the DNA repair. And essentially, at that time, there are some of the control mechanism, and the process is basically that if the DNA is not repaired in about 40 minutes in E. coli generation time is 20 minutes. So 40 minutes, essentially, if the DNA is actually not repaired, then there is a mechanism that is triggered, which we call as a translesion DNA synthesis. And at that time, what will happen, that not only these systems will try to repair the DNA, and if they fail, the DNA polymerase 4 and 5 will come in action. 4 and 5 doesn't have any proofreading ability like DNA polymerase 1, 2, and 3, and they are error prone. And they will synthesize the unrepaired DNA in this process. So again, in short, that the LEX-A destroyed, activate 50 DNA repair enzyme, and they will try to repair all the damages in the DNA. If it takes more than 40 minutes and the DNA is not repaired by these enzymes, then there is a second degree of is induced, which is a translation DNA synthesis is triggered, which will try to DNA damage by DNA polymerase 4 and 5. This will basically, even if you have an unrepaired DNA, they will just randomly put many sequences and try to do that. This can lead to hypermutation. Sometimes this could be lethal and still the organism may not survive, but it sometimes may survive. And not only it survive, sometimes what can happen, it can give rise to a drastic mutation, which can have many different types of implications as well. So this is the end of the part 1 of chapter 16.